In this video, I'd like to talk about neurophysiology, how the neuron works and how the neuron maintains itself at rest. And so to do this, what I'd like to do is first talk a little bit about the neuron at rest. Because the neuron at rest, it's a little bit deceiving term. It's not really resting, so to speak. It's actually hard at work. So its resting state is what's called the resting membrane potential. And so I want to tell you what that is and how that is established. And then after we go through that, number two, what I'd like to do is talk about a neuron at work. And when a neuron works, basically it can receive incoming stimuli in one area of the cell in a certain way, and then transmit that information in a different part of the cell in a different way. And so that's basically what I want to talk about in this video. So starting with the resting membrane potential, or the neuron at rest. Like I said, the neuron is not really at rest, it's in a state of readiness. And so I would make the analogy of a preheated oven. So when I preheat the oven to 350, it's in a state of readiness. When I need it to bake something at 350, it's ready to go. Otherwise, if it's not preheated, then, it's not in the state of readiness. But to have it in the state of readiness, it takes energy, right? So the same thing with the neuron. So the neuron, when it's not transmitting a message, neurons continually utilize energy to maintain this state of readiness, okay, kind of like the preheated oven. So in order for them to receive incoming information, that you need to be in the state of readiness. And that state of readiness is what's known as the resting membrane potential. And what it refers to really is this difference in charge across the neuron's membrane. So in this picture in the book, what they're showing us is a voltmeter. And you can see that they're placing one electrode outside of the neuron membrane and one electrode inside of the neuron's membrane. And so what the voltmeter is giving us is a measurement of the difference in electrical charge between the inside and the outside of the membrane. Okay, and because the recording electrode is being placed inside of the cell, what this tells us is that inside of the neuron's membrane, it's about -70 millivolts compared to the outside. So as far as what is the resting membrane potential, it's this difference in electrical charge. Okay, it's just a voltage reading. So typically neurons are going to rest around -70 millivolts, meaning that inside of the neuron cell membrane, it's -70 millivolts compared to the outside. And I explain that here in the notes. And so this separation of charge that we see is known as polarization. And so basically inside of a neuron, there are negatively charged proteins. These negatively charged proteins are going to attract positively charged ions to the cell. And so it makes sense that these positively charged ions are kind of hovering outside of the membrane. Also, it makes sense that since these positively charged ions are attracted to the cell, that negatively charged ions are going to hover on the inside of the membrane, kind of like a magnet, if that makes any sense. And so this is the polarization, the polarized membrane, where positive charges are hovering on the outside of the membrane and negative charges are hovering on the inside. And so actually, if you take a look at the extracellular fluid, it's electrically neutral. If we took a look at the cytoplasm, it's electrically neutral. But what we're seeing with this polarization, if you're familiar with chemistry, and say the structure of water, that the charge is being unequally shared. And so we're just saying that in this electrically neutral extracellular fluid, positive charges are more concentrated on the outside of the membrane. And the same thing here, that the cytoplasm, it's electrically neutral, but the negatively charged ions are in greater concentration inside of the membrane, and the two opposite charges are being attracted to each other across this membrane, and that's the polarization that's referred to as the resting membrane potential, such that the inside of the membrane is about -70 millivolts compared to the outside of the membrane. So the separation of charge can be measured with a voltmeter. The resting membrane potential equals approximately -70 millivolts, which means that the inside of the cell membrane is about 70 millivolts less or more negative than the electric charge outside of the neuron's membrane. It doesn't mean that the electric charge inside of the cell has an actual value of negative 70 millivolts. All this is a comparison. It just means that there's a 70 millivolt difference, and it's more negative 70 millivolts inside of the cell. So this resting membrane potential, you can imagine, unless you're using energy to maintain it, it will just dissipate, right? Because to have a difference over time, think of, say, like a heat in the house. To maintain a difference over time, you have to use energy. So if I want my house to be at 68 degrees and outside it's at 30 degrees, in order to maintain that 68 degrees over time, I'm going to have to utilize energy. It's also like the preheated oven idea. We're going to have to utilize energy. And so the resting membrane potential, the fact that it's negative 70 millivolts inside of the cell over time, it's using a lot of energy. And so in order to establish and maintain the resting membrane potential over time, these two types of channels are involved. And these channels are located all across the neuron. So in other words, whether I look at the dendrites or the cell body or the axon, teledendry axon terminals, no matter what part of the neuron I look at, there are these leak channels, which we're going to see leakage of ions using passive transport, meaning that they're just going to diffuse across their gradient. And then we'll also see a sodium-potassium pump all over this cell. And the sodium-potassium pump is an active transport pump. It's going to actually move sodium and potassium ions in the direction that it wants them. Okay, in other words, against what would happen passively. So let's take a look at that. So the active transport sodium-potassium pump, there's a picture in our book of it. So they're looking at this. They're using two pumps just to show us these two actions. But actually, each one of these sodium-potassium pumps will do both of these movements. It uses energy, it uses the energy of ATP to kick 3 sodium ions out and to take 2 potassium ions into the cell. So the active transport of sodium-potassium across the membrane, and remember these sodium-potassium pumps are located all over the cell, is going to create a concentration gradient. So check this out. Do you see how they're telling us that sodium concentration is high outside of the cell? It's at about 140 millimolars, and we don't have to know that number. And that it's at only 15 millimolars inside the cell, so the sodium concentration is low inside the cell. This makes sense because we said we're using energy to continually eject 3 sodium ions out of the cell. And so because we're using active transport to continually eject the sodium out of the cell, sodium concentration outside of the cell is high, sodium concentration inside of the cell is low. At the same time, this active transport pump will bring 2 potassium ions into the cell. So we're pumping potassium into the cell, and notice that the potassium concentration inside the cell is at about 140 millimolar. So there's a high potassium concentration inside the cell, low potassium concentration outside the cell. And so essentially what this active transport system is doing is maintaining this concentration gradient, but also maintaining that electrical charge difference over time. Now, across the cell, there are also leak channels. So these leak channels are always open. Okay, so they're always open, and there are both sodium and potassium leak channels. There's more potassium leak channels than sodium leak channels, so potassium is more permeable to cross the cell, but both of these ions can cross the cell. So just taking a look at this picture again, what does that mean? It means that sodium is going to use those leak channels to leak into the cell, but our sodium-potassium pump will use energy to eject that sodium back out. Potassium is a high concentration inside the cell, low concentration outside the cell. Potassium will continually leak out, but our sodium-potassium pump will use energy to take that potassium that escaped and put it back in. So do you see how that works? Across the whole neuron, sodium will continually leak in, and potassium will continually leak out. And the sodium-potassium pump will continually correct these movements by ejecting sodium from the cell and bringing potassium back in. And so over time, the sodium will use the leak channels to get into the cell. Potassium uses the leak channels to escape the cell, but that sodium-potassium pump will continually correct this movement. It's going to use active transport to move those ions in the opposite direction that they're leaking. So it's the sodium-potassium pump that's going to maintain this concentration gradient and this electrical charge difference, the negative 70 millivolts, the resting membrane potential, over time. I hope that makes sense. So essentially the neuron is using active transport to correct for these passive movements while the cell is at rest, and it will do that all over the cell over time. Okay, so let's move on to what's going to happen to disrupt the resting membrane potential. So the resting membrane potential can be disrupted. This just means that the cell is going to work. And so the cell, when it's at work, see if this makes sense. I made up this diagram to show that essentially two different things are happening. Okay, there's no overlap in these. And so unlike the resting membrane potential where we see that everywhere, we'll see that the cell can receive incoming information on the dendrites in the cell body. Okay, so on the dendrites in the cell body, the way that we receive that incoming stimuli is through chemically gated channels. And those chemically gated channels will allow ion movement that generate what's known as a graded potential. So the graded potential is just the way that the neuron receives incoming information. The other way that the neuron will work is like based on the strength of that incoming information, is it big enough to generate an action potential at the axon? That's essentially the question that we'll ask here. So if this incoming stimulus is big enough to generate an action potential, that is done on the axon. So on the axon, there are voltage-gated channels, and these voltage-gated channels can generate something called an action potential. I hope this diagram helps you to see that the neurons are going to do two different things in two different ways. We're going to receive incoming information through these chemically gated channels that generate graded potentials. Those graded potentials diffuse down to the axon where you could trip open a voltage-gated channel. And if you do, that will generate an action potential. Okay, so the voltage-gated channels are only located on the axon down to the axon terminals. The chemically gated channels are only located on the dendrites in the cell body. So let's look at this first by talking about how a graded potential is generated and what that is, and then ask the question of, will it generate an action potential and what is that? All right, so all of the gated channels that I'm talking about here, okay, the chemically gated channels in the cell body and the dendrites, and the voltage-gated channels in the axon terminals, all of those channels are passive transport. They're all going to work by passive transport. The only active transport that we're concerned with is this sodium-potassium pump that's maintaining the resting membrane potential. So these are just passive channels that open. And you can imagine, since we know what the concentration gradient is of sodium and potassium, if it was a sodium channel or if it was a potassium channel, we could predict what direction the ion's going to move and how it would change the electrical charge. So let's take a look at that. So with a gated channel, these gated channels that are located on the dendrites and the axon, the gated channels are closed unless a specific stimulus opens them. They're working by passive transport. In other words, when a gated channel opens, ions will diffuse across the channel. And it's going to diffuse across the channel according to the laws of diffusion, which we already know. So the ions are going to move from an area of high concentration into an area of low concentration. So the channels will be specific for a particular ion, either sodium or potassium. So chemically gated channels, these are the ones that are located on the dendrites and the cell body. They receive incoming chemical stimulation. And so when the chemical binds to a receptor, that receptor is not just a receptor, it's a chemically gated ion channel. So here they're showing us a neurotransmitter, that's that ACH, binds to a receptor, and that opens the channel. If this was a sodium channel, I think sodium will diffuse into the cell, knowing the concentration gradient, right? So knowing that sodium is high outside the cell, sodium's low inside the cell. If we opened up a chemically gated channel for sodium, I think sodium's going to enter. And because sodium has a positive charge, the inside of the cell is going to become more positive. If that chemically gated channel opened a potassium channel, potassium would leave, because potassium would move from an area of high concentration into an area of low concentration. And when a positive charge leaves, I think it would become more negative inside the cell. I hope that makes sense. We'll keep talking about this. The voltage gated channels are located all across the axon, starting at the axon's initial segment through the axon terminals. There are both voltage-gated sodium and potassium channels, and what they do is they open based on the voltage status. So here's a voltage-regulated sodium channel that I have in the notes. You can see the recording electrode is inside the cell. It's resting at negative 70 millivolts. That voltage-gated channel is closed. Now, here, the voltmeter is showing us that there's a depolarization. So take a look at that. I know it's small. It's negative 60 millivolts. Okay, so there's a depolarization. So in this case, that negative 60 millivolts must be the threshold depolarization, because I see that the voltage-gated channel has opened. And because I know this is a sodium channel, sodium's going to move from its area of high concentration into its area of low concentration. It's going to move into the cell passively. And then with a voltage-gated sodium channel, these channels also inactivate before they close. So here, the voltmeter is saying positive 30 millivolts. And around positive 30 millivolts, that's when voltage-gated sodium channel inactivates. So much sodium, enough sodium came into the inside of this membrane that this voltmeter sees that it's positive 30 in here compared to the outside. And at about that time, you can see how this inner gate closed. That's what's known as inactivation. Eventually, this voltage-gated sodium channel would go back to closed. Okay, so it would go back to closed. So if we depolarize it to threshold, if we depolarize it a certain amount, or I should say change the electric charge inside a certain amount, that causes it to open. And then another electric charge is going to cause it, so when a certain amount of ion comes in, it'll inactivate, and then it will eventually go back to closed. So that's how the voltage-gated sodium channel works. And the voltage-gated sodium channel, that's what you see on the axon. This is going to generate an action potential compared to the chemically gated channel. That's going to generate a graded potential. So in our resources, there are two interactive physiology activities that I'd like you to do. They're in the Brightspace folder. I'll talk about them in the announcement, but these videos just link right to those videos, those animations, which I think you should look at. So as far as changing the resting membrane potential, let's take a look at that. So to change the resting membrane potential, we can see some ways that that could happen. You know, it could happen if one of these gated channels opened. So if a chemically gated channel opened or a voltage gated channel opened and ions moved in or out of the cell, that would certainly change, you know, that negative 70 millivolts. It would become less stable, right? We would get a different reading on the voltmeter. So in this picture, what they're showing us is that a stimulus was given at this point in time. And I know that because I see it resting at negative 70 millivolts up until that time. And then I see that the cell's becoming more positive. You see how it didn't go all the way up to 0. It didn't even get to negative 50 millivolts, but it's becoming more positive. And when it gets more positive, what happens is the difference between the inside and the outside of the cell. So here's the difference. If we went up to 0, all that means is there's no difference in electric charge between the inside and the outside. Right now at rest, it's negative 70 millivolts, more negative. It's more negative inside the membrane than out. But you see with this depolarization, they're calling this a depolarization because the amount of difference between inside of the membrane and outside of the membrane has lessened. So we know that to have that difference, that's what's known as to be polar, right? That's polarization. That's a polarized membrane. So I hope it makes sense that if the inside of the cell becomes more positive and it lessens that difference, that's a depolarization. So a depolarization inside of the cell is when the cell becomes less negative or more positive, however you want to think of that. There's a reduction in the difference between the electric charge inside the cell with the electric charge outside the cell. So you might wonder, what would be a typical cause of a depolarization? If a chemically gated or voltage gated sodium channel opens, because we have a high concentration of sodium outside the cell and a low concentration of sodium inside the cell, doesn't matter how the channel is gated, right? If it's chemical or voltage, if you open that, sodium will passively enter the cell. Sodium carries a positive charge, which means that it's going to make the inside of the membrane more positive. And that is what we call a depolarization. Okay, so that depolarization is most likely due to sodium ions entering the cell through a gated channel that was opened. You could have the opposite happen. You could have a hyperpolarization. And a hyperpolarization is when the inside of the membrane becomes more negative, less positive. So there's an increase in the difference. And I'm just going to make this a little bigger just for a second to mark this up. So the cell is resting at negative 70 millivolts, and I can see right here it changes. So there must have been a stimulus that was given at that point. And I can see that the cell is becoming more negative. So now it's going from negative 70 towards negative 80, right? It's becoming more negative. So we know at rest, the polarized membrane, there's a 70 millivolt difference. Now, what's happening is the inside of the membrane is even more negative. And because it's more negative, we'd say that's hyperpolarized. The difference is even bigger. So with hyperpolarization, there's an increase in the difference between the electric charge inside and outside of the cell. Okay, there's an increase in the membrane potential. What would be the typical cause of hyperpolarization? That we opened up potassium channel. So if we open a potassium channel, whether it was a chemically gated or voltage gated potassium channel, here I'm just using this picture to look at the gradient. We know that potassium is higher concentration inside the cell, lower concentration outside the cell. So if I open a channel that allows the passive movement of potassium, potassium will leave. And when a positive charge leaves, it becomes more negative inside of the membrane. And that's what we see. So it's becoming more negative inside the membrane. So the typical cause of hyperpolarization is passive movement of potassium. Potassium channels open. Repolarization just means that as long as a cell is living and able to use energy, that we can use and will use the sodium-potassium pump to restore the resting membrane potential. And you see that here. So even if the cell became more negative, it's the sodium-potassium pump using ATP to eject the sodium and reclaim the potassium that will reestablish the resting membrane potential. Let's look at this one. So here, the cell was at rest, and then it depolarized. But then this little segment of the graph is what we would call repolarization. Repolarization, this is where the sodium-potassium pump is reestablishing that resting membrane potential. So we're using energy to keep it at that negative 70. So here in the notes, what I do is go through the steps of first how the neuron receives an incoming message as a graded potential, and then ask the question, could it generate an action potential on the axon? So graded potentials, remember, this is how the neuron receives incoming information. It's short term. It's just a temporary change in voltage. It only occurs in the dendrites and the soma. And then what it will do is this electrical change will diffuse towards the axon initial segment. Or in other words, it's going to diffuse towards where the axon first starts. And the thing is, because this is just a little local event, it gets weaker as it moves. So a great analogy for this is like a ripple in the pond. So, you know, it's strong where it first occurs, and then as it radiates out, like towards the axon, it gets less and less. So I wrote down all of the steps, but I'm going to take a look at a picture with you. Let's mark up this picture, and then we'll go back to the steps to make sure that we have everything written down. So here they're just showing us that a stimulus was given at a point on the cell body close to the axon. So if we're saying that the axon, I'm going to just draw a line there, starts here, basically what you're saying is that you have chemically gated channels here that can generate graded potential, and we have voltage regulated channels on the axon. So what they're showing us in this picture is they're showing us what's happening on the cell body. So do you see how this is a chemically gated channel? This is a chemically gated channel. Okay, so it has that little receptor site for the chemical. And they're showing us that the cell is at rest. It's at negative 70 millivolts. They're also showing us how the sodium concentration is high outside of the cell, and that the inside of the cell is negative 70 millivolts compared to the outside, so the cell's at rest. Next picture, a stimulus. The stimulus is the chemical. So this chemical that they're showing bind to the receptor, this chemical is likely a neurotransmitter. So when a neurotransmitter is released, that neurotransmitter is a chemical that's going to bind to the chemically gated ion channels. This is a chemically gated sodium channel. So we can see now that it's bound, the chemically gated channel opens. And because it's open, what's happening is that sodium is moving from its area of high concentration into its area of low concentration. So sodium ions are physically entering the cytoplasm of the neuron. And as these positively charged ions physically enter, look what happened. The inside of the cell became more positive. So it went from negative 70 millivolts to negative 65 millivolts. And so taking a look at the last picture, what they're trying to show us here is that where you open the channels, so we open the channels here, and so we're getting sodium diffusing in, and I'm getting a big difference in electric charge here because so much sodium has come in that it's made the electric charge go from negative 70 millivolts to negative 60 millivolts. And of course those sodium ions, they're not going to, you know, be rigid and stay right there in the cytoplasm. They're going to diffuse from their area of high concentration to their area of low concentration. So here, where they show us a local current, all that local current means is the diffusion of sodium, the diffusion of this positive charge. So do you see how when some of these positively charged sodium ions diffuse into the neighboring area, look what happens. It's going to change the membrane potential, but not as much. So here, you know, it was resting at negative 70. Now it's at negative 65. Where I open the channel, it's at negative 60. But you see where no sodium has diffused into this area, that the membrane's still at rest. It's still at negative 70 millivolts. And so the way they're showing it on this picture is like a target, you know, that you're going to have like the biggest effect where you open those channels. I'm going to color code this. And then that is going to radiate out, you know, some ions are going to diffuse from that area. So you'll still have a difference in electrical charge, but it just won't be as much. And, you know, so on. So here's like the lighter area. So it's just like the ripple in the pond, it's dissipating. But the question that we're asking is, will there be a depolarization that enters into where the voltage regulated sodium channels are located that's big enough to open those voltage regulated or voltage gated sodium channels? If so, that stimulus was big enough to generate an action potential. If not, that graded potential, okay, what the stimulus generated, just stops right there. So taking a look at those notes. Neurotransmitter, so this is the chemical, binds to the receptors on the dendrites and the soma. This is the incoming stimulus. The neurotransmitter receptor is part of the ion channel. Channel is closed, but when the neurotransmitter binds, it opens the channel. These channels are going to be specific. They're going to be either sodium or potassium channels. Once a channel is opened by a neurotransmitter, ions will move passively by diffusion. So it's possible that a sodium channel would open, and if a sodium channel opens, which way would sodium move? Sodium would enter the cell, right? So sodium would enter, and if a positive charge enters, inside of the cell is going to become more positive. That difference is going to not quite dissipate, but lessen. And so we call that a depolarization. Okay, if a potassium channel opens, we have a high concentration of potassium inside the cell, low concentration of potassium outside the cell, and so we would expect that potassium would leave. And if a positive charge leaves, that means that inside of the cell, it's going to become even more negative. And that is known as a hyperpolarization. So next, what we'll see is there's either going to be a depolarization or a hyperpolarization. It depends on what type of channel opens. But we know that the magnitude of that voltage change is going to depend on the number of channels that open. So did a lot of sodium come in? Did a medium amount come in? Did a small amount of sodium come in? To use my rock in the pond analogy, did we drop a boulder into the pond where you're going to have huge ripples that like radiate out far away from there? Or did I drop a pebble into the pond where, you know, that's going to dissipate right away? So that's what is meant by a local event. It's strongest where the channels are opened, and then it radiates out from that area, which means that it could reach the axon initial segment. And this is what we're waiting for. If it's a depolarization, and if that depolarization reaches the axon initial segment, it has the potential to open voltage-gated channels that are located there. What would have to happen is we'd have to depolarize the axon to what's known as a threshold level. The threshold level would be the electrical charge that causes the voltage-gated channels to open. All right, so that's how the graded potential works. Now with the action potential is only generated by the axon. The dendrites in the cell body can't generate action potentials, and the axon doesn't generate graded potentials. Okay, so I hope that makes sense. I tried to show you that with that two-colored neuron at the start of this. So an action potential is the same thing as a nerve impulse. It's an electric current that can be generated by the axon, and if it is, it will be conducted down the length of the axon without getting weaker. And so an action potential is very different in that way. It's almost like dropping the match on the gas line. If you generate an action potential, it won't get weaker and it will be conducted down the entire length of the axon, which is going to cause a neurotransmitter to be released from the axon terminals of that cell. So this is the way that a message gets communicated between neurons. And so we can talk about an action potential in different phases: resting, depolarization, and repolarization. I'm going to look at a picture, go through that, and then actually, let's look at this picture first. Let's take a look at this graph, because this is a single action potential. In other words, what this means is that we're just looking at one piece of the axon where that electrode is, recording, where it was poked into the axon, and it's comparing still to what the electric charge is outside. So let's take a look. So here, we can see the axon is at rest, and here's a depolarization drifting in. What is that depolarization? It's a graded potential, like we just talked about. It's a change in the charge that was caused by chemically gated channels in the dendrites in the cell body opening, causing sodium to physically enter the cell. Those sodium ions, positively charged ions, physically diffuse into the axon. And if they reach a threshold level, voltage gated sodium channels open. And when the voltage gated sodium channels open, sodium comes into the cell. This is that depolarization. So when you open the voltage gated sodium channels, that positive charge is hovering the cell. It's electrically attracted to the cell. There's a concentration gradient. Sodium's high outside the cell, low inside the cell. And so, you know, when you open that gate, sodium's coming in. And so much sodium, enough sodium comes in that the inside of the membrane actually goes to positive 30 millivolts. And at that point, this is when two things happen. One is, I showed you this earlier, the sodium channels inactivate, so they won't let any more sodium in. But the other thing that happens is this is around the time that the voltage-regulated potassium channels open. So we have both voltage-regulated sodium and potassium channels. The potassium channels just are kind of slower, okay? They're slower. So when I trip open the voltage-gated sodium channel, it's going to take a millisecond for the potassium channel to catch up. But when the potassium channel opens, the voltage-gated potassium channel, there's a very different story of what will happen, right? Because the potassium concentration is high inside the cell, low outside the cell, so we expect potassium to leave. Furthermore, because we let all of the sodium in, now the inside of the membrane is more positive than the outside of the membrane, so not only does the concentration gradient of potassium drive potassium to leave the cell, but potassium, positively charged potassium, is electrically repelled by all the positively charged sodium that entered the cell. And so if you open these voltage-gated potassium channels, potassium leaves. And whether you say voltage-regulated or voltage-gated, it means the same thing. I used voltage-gated in the notes because that's what our book is using. So potassium leaves, and when a positive charge leaves, cell becomes more negative. So we see that. That's a repolarization. And right around the time you get to like the threshold level or resting membrane potential, that's when the voltage-regulated sodium channels will start to close and also potassium channels close. But just like we saw with them opening, the sodium channels will close first and then potassium are slower. And because they're slower, do you see how potassium is going to continue to leave until all of those are closed? And once all of those are closed, what will happen is the sodium-potassium pump, like right here, it's going to bring it right back up to resting membrane potential. It's going to reestablish the ions on either side of the membrane. So I wrote this out here. So the resting state is that all voltage gated channels are closed. In order to generate an action potential, all that needs to happen is you need to open these voltage gated sodium channels in the axon initial segment. That's just the first part of the axon. So this is, you know, where the sodium ions would diffuse into from the cell body and the dendrites. So these channels will open if voltage changes from the resting membrane potential, negative 70 millivolts goes up to about negative 55 millivolts. Our book says negative 55. I know the pictures that we're using say negative 60. This, you know, is a ballpark number. It varies from, you know, cell to cell. So please don't worry about that. I guess the main thing to know is it's a threshold stimulus. So if we change that to threshold, so if we depolarize at about 15 millivolts, 10 to 15 millivolts, the voltage-gated sodium channels will open. The threshold stimulus is produced, remember how this happened, by a graded potential that was large enough to depolarize the axon. In other words, chemically gated channels, enough of them opened out in the dendrites in the cell body that allowed enough sodium ions in that they drifted towards the axon and depolarized the axon to threshold. And when that happens, voltage-gated sodium channels open. So this threshold stimulus is a little bit like pressure on the trigger of a gun. This is a good analogy. If you open those channels, that's all you can do. So in other words, if I, with pressure on the trigger of a gun, if I like very slowly, you know, meekly, you know, softly press the trigger, as soon as you reach that critical pressure, the gun's going to fire. And it fires in the exact same way. It doesn't fire light if I press really lightly on the trigger, or if I like slam down the trigger with heavy pressure, it doesn't fire stronger. That's what an action potential is like. All you need to do is depolarize the axon initial segment to threshold with the sodium that is diffusing in from the graded potential. So it doesn't matter if you like very slowly, barely make it there, or if like you drop the boulder in the pond, if lots of sodium comes in there, same outcome. You open the voltage regulated or voltage gated sodium channels. And in that way, action potentials are yes or no. They're all or nothing. There's no such thing as grading it small, medium, and large. It's all or nothing. If you open that voltage gated sodium channel, you have an action potential. So we'll continue to look at that. Not every stimulus will. So a sub-threshold stimulus would be a depolarization that falls short of opening the voltage-gated sodium channel. So using 55 as the threshold, do you see how if I got a little bit more positive than -70, but I didn't make it to -55, that would be a sub-threshold stimulus. So in other words, you didn't reach the critical pressure on the trigger. Nothing happened. Okay, so in which case the voltage-gated sodium channel stayed closed and no action potential occurs. But if the voltage-gated sodium channels are opened by a threshold depolarization, we know what happens here. I showed you this on the graph. Sodium will enter the cell. That's going to cause a depolarization to about positive 30 millivolts, which just means that the inside of the membrane is about more positive 30 millivolts compared to the outside. And then at about positive 30, two things will happen. We said that the voltage-gated sodium channels will inactivate, so sodium can't enter any longer. They're not closed, so to speak, and this is an important point that we're going to build on next week, but we're not letting any more sodium in. And then the voltage-gated potassium channels open more slowly. So once they open, potassium is going to leave, and that's going to cause the inside of the membrane to become more negative because positive charges are leaving, and that's our repolarization. So around resting membrane potential, the voltage-gated sodium channels will close, and the voltage-gated potassium channels will close, but they close more slowly, so some close, some stay open, and as long as you have potassium channels that are open, potassium will continue to leave, a further potassium efflux, and that causes characteristic hyperpolarization at the end of an action potential. So once the voltage-gated potassium channels close, leaving all the voltage-gated channels closed, what would happen is the sodium-potassium pump will use active transport to reestablish the resting membrane potential. So when you take a look at a graph like this, what we're really looking at is where the electrical changes that have occurred right where the recording electrode is in the cell. So in other words, just to take a look at a picture, let's look at a simple picture like that we started with. This picture. So here, very simple picture, they're showing us that they're going to record the electric charge inside of the membrane and compare it to the outside of the membrane, and we get the readings here. When voltage-gated sodium channels in this patch of membrane open and sodium comes in, this is when you're seeing the positive 30, and then those inactivate, and then the voltage-gated potassium channels open, potassium leaves, and then it goes back down. You see the hyperpolarization, then it reestablishes. So I hope that makes sense. So that graph that you're looking at is really just the changes that occur, basically like this reversal in the polarization at that exact spot where the electrode is placed. And so in other words, if we saw an action potential here, then if the electrode was any other place, that electrode would be at -70. But as I was mentioning, once you generate an action potential, it will perpetuate itself down the length of the axon. And I just want to look at that to finish up this video. So I put one other picture in the notes, this one. So in this picture, what we're looking at now is we're focused in on the axon. So they're showing us the axon. They're showing us the voltage regulated or gated sodium channels. They're showing us the voltage-gated potassium channels, and they're showing us a graph on the side. So here we can see that the axon is resting at -70 millivolts. Next picture, this would be like the color-coded graded potential. Remember I darkened this like red. So this is where the chemically gated channels open. And then this is where some sodium is diffusing, less sodium is diffusing. But we can see that the sodium is diffusing into where the axon starts, which this is the axon initial segment. That's going to cause a depolarization because it has a positive charge. And so what do we see? We see you go from rest to becoming more positive. So all you're waiting, like I said, like pressure on the trigger of a gun, is will these sodium ions that diffused into the axon depolarize the axon to threshold, opening the voltage-gated sodium channels. And of course, you know, in this picture it will, because otherwise there would be no use of looking at this picture. So, so here they're saying it did depolarize to threshold. So right at that point, voltage-gated sodium channels would open. So I can see these voltage-gated sodium channels open. And then from here, it's passive. Keep that in mind. You know, so because we have a lot of sodium outside the cell and not as much, a lower concentration inside the cell, what happens is sodium moves through these open voltage-regulated sodium channels, and as that positive charge enters, you know, surprise, look what happens. The cell becomes more positive. It goes up to about positive 30, and at positive 30, what we see is that the sodium channels are inactivating. So that's that inner gate. You know, compare that to out here where we were saying it was closed. That's how they're just depicting inactivation. So the sodium, the voltage-gated sodium channels inactivate, but the potassium channels now open. So now with the potassium channels being open, potassium's going to move passively. And because we have higher potassium inside the cell than we do outside the cell, plus inside the cell is more positive, potassium's definitely on its way out. So potassium will just passively leave through these open voltage-gated channels. And when a positive charge leaves, we can see what will happen. It's going to become more negative. Now, this is considered a repolarization because it's going back to resting membrane potential. So around threshold resting membrane potential, this is where we see the sodium channels close. So they go from inactive to officially closed. And then look at the potassium channels. Do you see how they're showing us one is open, one is closed? So they close more slowly. And if it's open, potassium continues to leave. And that's why we're getting this hyperpolarization, because we're getting the additional positive charge leaving. Yeah, so that covers the action potential. So we'll talk about how it propagates down the axon in the next class, next video. Thanks a lot.